Good day, everyone. This presentation, I will just upload it quickly. Right, thank you for the opportunity to make this presentation. It is not Dion Mostert that will be making it. Rather, I will do this in collaboration with Megan Matthews, that's also working in the group. And the presentation will be divided in two different aspects. Firstly, I will talk about the fungus in generally and how to identify FOC. And then Megan will continue with the detection of the fungus inside environmental matter, such as soil, water, and so forth. And just to start by explaining this very complex fungus, I want to say that Fusarium oxysporum can cause a disease to a number of different crops, about 120 to 150 different ones, all getting Fusarium wilt diseases. And these diseases are then caused by different strains that we usually refer to as forma specialis of Fusarium oxysporum. The forma specialis indicating a special form that will affect banana, or it will affect tomato or cotton or other plants. So strains that will be affecting banana would not affect the tomatoes, or it will not affect cotton. So it's very, very specific. There are some exceptions, but generally these strains are very specific. And this is what the fungus looks like. It's got some very characteristic structures that they produce. The three different types of spores. Firstly, the macroconidia, which you can see over here. Then the microconidia, and these microconidia are very often the most abundant of all, and they are produced in these falsets on short conidiophores. This is a very typical characteristic of it. And finally, you get the chlamydospores, and these chlamydospores will form either singly or in pairs, and they can form either inside the hyphae or even inside the macroconidia, like this one when they age. These are the spores that survive in the soil later on to infect new plants. They're very hardy, and they can survive up to 30, 40, or 50 years in the soil. What is important to recognize from this morphology is that the morphology of all the different forma specialis of Fusarium oxysporum looks the same. They are not different. So if you look at something affecting tomato, or you look at something that's affecting cotton, the spore types will look very much the same. There is, however, some variation that you will find, but that variation even occurs within the forma specialis. That you will find that they, for instance, produce a different color when you grow that on an artificial medium, or that the spores might have different numbers of septa. So it's a natural variation that will occur within a specific group of the fungi. It is not linked to a specific forma specialis. Most of the Fusarium oxysporum that's found in the world, and they are found very widely, absolutely everywhere where you can find them, are non-pathogenic. So you will find some in the soil, and you cannot directly say that they are causing a specific disease of some type of crop. And they are just occurring there in the soil naturally, or have been introduced there. But these then might become pathogenic under certain conditions. And two of those conditions that we speculate is the reason for these non-pathogens moving into a pathogenic strain, or when you get the selection culture or the selection pressure of monoculture production on Fusarium, non-pathogenic Fusarium oxysporum. So there are mutants occurring, and that then become pathogenic to the crop, or that you get horizontal gene transfer. This has been demonstrated more recently, and there's good evidence that in Fusarium oxysporum, this is quite a common feature, and that might result in pathogenic strains developing. So most of the strains, if you look at this figure over here are saprophytic. They're also asexual, so they do not diversify by means of recombination. It's either by small mutations or by this horizontal gene transfer. And in this way, the non-pathogenic strains might become either pathogenic to tomatoes, and we will then call them Fusarium oxysporum formis specialis lycopersicii, or melons, and that's got a new name called Fusarium oxysporum formis specialis melanus. And then obviously the one causing disease to banana is called Fusarium oxysporum formis specialis cavendish. This specificity in its pathogenesis is due to a very specific feature within the genome of Fusarium oxysporum. It basically consists of two groups within the genome, one with conserved functions that we call the core genome, and then the other group of set of chromosomes that is responsible for pathogenicity, and this is what we call the accessory genome. And it's important to realize this once we start working with the identification of the fungus. There are also a number of different methods to identify Fusarium oxysporum, and many of these have been used over the years. Some of them are not that common anymore, but they can still be used for identification. In terms of cultural characteristics, one has to look at the growth rate, for instance. Also the mycelium morphology. What we find with Fusarium oxysporum is that it does show a cottony type of growth. It has this purple center very often, and that center can be quite small, but it might also be larger. And then it's got a general cream color from the top and from the bottom. Morphological characteristics are the microconidia and the macroconidia and the chlamydospores and their presence. Also sporodokia and the conidiophores, which in the case of Fusarium oxysporum are monophyletes, which is very important in the larger genus of Fusarium to understand how to differentiate between the different species. When you phenotype it, firstly one has to look at the strains that they have to be pathogenic to their specific crop. So if they are pathogenic to banana, they will be Fusarium oxysporum formis specialis cavendish. And then you can also find races, and races are defined based on the different cultivars that they will affect. Vegetative compatibility groups are really important, and I will explain a little bit later about their importance. And then volatile production that was used previously to separate some strains from others, simply by the volatiles that are being released by the fungus when grown in culture. And finally, there are different ways of molecular identification that I will also discuss just now. We all know that banana Fusarium wilt started to become a problem internationally in Latin America, where it affected Gros Michel very severely, and that Gros Michel was eventually replaced by Cavendish bananas, which showed complete resistance to the strain of Fusarium oxysporum cavendish in Latin America. And this Cavendish then later became susceptible to Fusarium oxysporum cavendish in other parts of the world, including Asia, where it caused severe destruction of banana monoculture Cavendish banana plantations, first in Indonesia and Malaysia, but it also spread later to other parts. And this made people realize that races are involved and that it's not a single strain that's responsible for the disease to banana. And generally, those races can be divided in 3 different groups, and I here just bulk the FO race 1 and 2 strains because on the gene level they are very similar, even though they do affect different cultivars. And then the Fusarium oxysporum subtropical strains and the Fusarium oxysporum tropical strains, tropical race 4 strains that can affect Cavendish bananas in subtropical conditions and in tropical conditions. And when you compare the genome, there will be differences between these 2 different groups. Fusarium tropical race 4, TR4 then became the real severe threat to banana production globally. It was first found in Asia, then spread to the Middle East and Africa, spread further in Asia, and is now also found in Latin America, as we all know. Then vegetative compatibility groups. And this is a very important aspect to us still. It is not something that can measure genetic distance between strains, but it can recognize similar strains, ones that are absolutely identical to each other. And that happens through a process of heterokaryosis and recognition over there. And that helps us to actually map and look at the spread and the diversity of this fungus around the world. This is one example. If we look at the diversity of Fusarium oxysporum cubensi in Asia and in Indonesia particularly, one can find there's a great number of different VCGs present over there. And this indicates also to us that the fungus has probably evolved with its banana host in Southeast Asia simply because of the great diversity of bananas that's present in the area. And one can now trace how these VCGs will move to other parts of the world and see if something is present in Indonesia, for instance, and you find the same ones outside the Asian continent, then maybe there is a link between these different strains, but they are similar because they are basically very close to clonal. The same work was done in Africa, where we did a large-scale mapping of VCGs in East and Central Africa. And we found that there are about 6 different VCGs present in this area. They all belong to a specific lineage. They are all closely related genetically, but they are different VCGs. And we have also seen now that one of these VCGs are starting to dominate in Africa, which is VCG 1222. If we were not able to identify these different VCGs, we cannot see these dynamics in the population structure of the fungus as it moves into a continent. And then, as I mentioned, some of these VCGs are quite closely related genetically, and then they are grouped into what we call lineages or different groups, which are phylogenetically related. And over time, people have identified between 5 and 10 of these lineages, depending on which technology you use or method you use, and also depending on the number of isolates that's being included into the study. But generally, there's about 7 or 8 of these lineages which are very common and divide the different VCGs into these groupings. I finally just want to talk about the accuracy of FOC diagnostics. It is really important that once you identify the fungus, that it's accurate and that you do not cause any panic in a specific area by doing false identifications of the fungus. And there are different ways of it, and obviously one wants to do it as rapidly as possible, but it has to be accurate as well. So molecular markers are the one tool that can help you to identify the fungus within 1 day. But if it's wrongly identified, then you have a problem with that. And some work has been done by Freddy Makdama in Ecuador, and he's still continuing by comparing some of these different tests that's available, which is really important for us to know which ones are available to use when you have to do identifications. I want to summarize the identification of FOC with this slide by saying there are important qualities needed for FOC diagnostics. Firstly, it has to be accurate. Otherwise, you do not really do what you are supposed to do. And it's also important that if you find TR4, for instance, in a new country, that you also use more than one test to identify that fungus and not just report it on the first test that you are doing. It has to be rapid. One cannot wait 3 weeks or 2 months to identify something. Some of these tests are taking much longer than others. For instance, VCG testing is not a quick test. It has to be affordable. So people in developing countries must also be able to use the technologies and afford the technologies in their countries. It must also be user-friendly, so not too complex for people to use. And finally, the identifications should be safe and secure, so one cannot just move cultures around the world indiscreetly. One has to basically respect the quarantine measures and the phytosanitary measures in the different countries. So one has to make sure that you can move something which is safe to move to another country for identification if needed, or you have to identify it in your country, at least provisionally using PCR methods where you only use molecular and DNA-based technologies. Right, aspects of FOC identifications that's really important. Firstly, is field symptoms. So if bananas turn yellow in a typical manner that Fusarium wilt might cause disease, one has to be alerted by that, and one has to collect some isolates to confirm in the laboratory as quickly as possible. It's going to be a very different and difficult task if one simply want to go to non-symptomatic plants in large plantations and start collecting isolates randomly. It will become a very expensive exercise. Once you're in the laboratory, it's also important to work from single spore isolates. I know a lot of people are still using just the culture that's growing out of the sample or the plant tissue, but it is also important to know that Fusarium, it's important to single spore these to make sure that you do identify the correct one that you try to identify. And finally, some pathogenicity testing that needs to take place, just to confirm that the isolate is not a non-pathogen, but indeed a pathogen that you're testing. I mentioned some laboratory identification methods before, the volatile production that is still being used by some groups, but not commonly anymore. Vegetative compatibility group analysis, that's really important, especially if you want to work on population and try to understand the movement of this fungus in the regions. And finally, molecular identification that can consist on PCR or qPCR techniques, and you can either work on housekeeping or effector genes. And then finally, sequencing that's become very powerful these days when you can do a genome sequence or even dot sequences to compare the different strains. And with this, I will now be handing over to my colleague, Megan. So Megan, if you would please.